Sam already introduced me very well. My name is Malaika Ebert. I started as the dry bean and pulse crop pathologist here at NDSU in the end of 2021, I think. So with me taking over the dry bean and pulse path lab from Dr. Pashi, also a couple of things change in the way we do research. So my lab tries to connect fundamental and applied research. So we use a lot of cutting edge technology to actually feed into the needs of applied research. So our research is kind of standing on two pillars. Both of them are equally important. One thing that we are continuing from what has been done for probably a decade already is the dry bean disease survey. And last year, we went out 3 time points. Once in the middle of July to score or look for root rots in the fields. Then at the end of July, beginning of August, that's when we go out to look for bacterial diseases and survey different fields in North Dakota and Minnesota. And then in the middle of August, we went out one more time to look for rust, white mold, and anthracnose. So what we find, we went to roughly 40 fields, and we found that, as always, root rot is very prominent in the fields. That is not a surprise. It's been like that for a couple of years. And as equally prominent are bacterial diseases. So we find them quite a bit. We did find a little bit of white mold, and I'm happy to report we did not find any rust. However, rust is something you should always keep in mind when the conditions are right. Rust can emerge very quickly. And then also, we did not confirm any anthracnose. So when we score for anthracnose, we look for symptoms. And once we find plants that we're like, maybe that could be anthracnose, we will take them with to our lab and analyze them molecularly, just to make sure that we are actually what we're working with. So we did that for everything that we sampled for anthracnose, and I'm happy to report that none of them turned out to be anthracnose. Because there's a lot of Fusarium root rot, I mean, you all know that. This is one of the major research points in my lab for Fusarium. This is a picture of, where's the pointer? Here. This is how it looks like when we take samples from the field back to the lab. We take the whole plant and we try to get as much of the roots as possible because they help us to get the pathogen out of the plant. You can see that most of those plants are very stunted, and you can see also that the root biomass is not very high. So when we look at this disease scoring here, this is a healthy plant, you know, everything's green and there's very thick root and with a lot of root hairs and all that. And with progression of the disease, you can see that the symptoms become more and more prominent. We get reddish, brownish discolorations, and also the amount of roots start getting lower and lower. These plants here, we will disinfect, and then we kind of trick the fungus to come out of the plant, and then we can keep the fungus. So when we talk about Fusarium root rot, I know Fusarium is usually something that sounds like a unit, right? But when you look at Fusaria in general, they all look a little bit different. So all of these plates that you see here are some sort of Fusarium, right? They differ in color, they differ in size, they have a lot of things, distinctive differences that we use to tell them apart. And in our fields, the 3 common species that we find are Fusarium oxysporum, Fusarium solanae, and Fusarium acuminatum. So all of them belong to the genus Fusarium, but they do have different features, if I may say so. And I think it's interesting to share with you how we determine what kind of fusarium we find in the fields. So, as I mentioned, these are the three most prominent that we have. But with once we put a name on them, that also tells us a little bit of how this fusarium can infect the plant. So they all have like little um, weapons, or they have different ways to infect the plants. And by looking at the species, or by knowing the species, we have a rough idea of how this fusarium can actually make the plant sick. So this is also why it's important to put a name on the pathogen. And we do it for one by looking at the plates. You saw some of them have a little bit, they differ in color, they differ in how fluffy they are. That helps us. But the most precise way to determine what kind of fusarium it is by molecular analyses. And for that, there's actually those fungi, they have a pretty cool feature. They have one tiny part of them that pretty much acts like a barcode. So all of them have those. And when you have a scanner, you're able to scan this little barcode, and then your scanner tells you what kind of species it is you have there. And that basically also tells you what kind of weapons you can expect from this fusarium. So in our case here, we scan the barcode, and the scanner tells us, oh, this is a Fusarium oxysporum. And then we note that. And then we have another Fusarium species. We take the scanner, we scan them, and here it says us, oh, look, this is a Fusarium solani. And this is very important. So we keep the cultures and we write down what name it is. But the other thing that is very important is how aggressive are those species on dry beans. And all of the species that we sampled from the survey, we make sure that they're clean, and then we run them in greenhouse assays. And there we kind of challenge different bean species with different fusaria. You can see a control plant here. This is just inoculated with water. This helps us to determine how the plants look like when nothing is threatening them. And this is always the set that we compare everything we inoculate to. Here you can see that the plants all germinated. They all have pre-big leaf. They're pre-green. However, when we challenge them with different fusarium species, you can see that they don't do very well. A lot of times when the fusarium is very aggressive, there's nothing germinating at all. So we don't even get like a little planty. Sometimes one or two germinate. Sometimes they germinate, but they're stunted. They come in all kinds of different shapes. It depends on how aggressive this fusarium is eventually. For our research, we needed a fusarium that had specific features. And in our case, this was this fusarium. We liked that it actually allowed plants to germinate, because that is what we need when we score for new varieties. We cannot have everything dead, because that gives us nothing to score by. So we took this fusarium that, according to the barcode, was a Fusarium solani. And unfortunately for this fungus, I like to look into things very deeply. I like to know what I'm actually working with. And to do that, we looked at all the genes of this fungus. So we not only looked at this little barcode, but we looked at the whole thing very, very closely. And that is when we noticed that although the area where the barcode is sitting tells us it's a solani, it is actually not. The rest of the fusarium doesn't look like it, which also tells us we don't know what kind of weapons it has. This is not concerning at all. I don't want to worry you. I just want to tell you what technology actually can do. So the take-home message for the fusarium project that we're currently working on is that, you know, as in other parts of life, things are not always what they seem. And it also tells us that we should keep an eye on the pathogens and how they adapt in their environment, because sometimes you get surprises. We will use this one because we don't know what it is exactly to identify the little weapons. So we can give the breeders also a better picture of what kind of weapons this fusarium might be using to infect the plant, and the breeders then can translate these things into what do I need for a line to become resistant. This is one part of the fusarium things we are doing, but also not only fusarium are very prominent, also bacterial diseases. And for that, we have three common ones. So this is common blight, brown spot, and halo blight. And I'm pretty sure Sam can tell you a little bit more about what makes bacterial diseases hit very hard. It's a lot of hail and wounding of the plants. So these are the three common ones that we're working with in the lab. So also during the survey, the survey is very important to us because we get all different new isolates that we find in our fields, right? And also those bacteria we will start to score like we do the Fusarium. We need to put a name on the disease in order to be able to handle it properly. And with the bacteria, we like to do different plate assays, we call that. So we grow the bacterium, and depending on how it looks like when it grows, we can tell what kind of bacterium it is. For example, the medium that is growing on is white. It has casein in it. There's also the stuff that makes milk white. You can see that this this bacterium, for example, does not do anything with a white background, right? It just grows there, it is in color. So we can tell this is a Pseudomonas syringae fasciicula. However, when we see, oh, look, this bacterium looks a little bit yellowish. It actually looks lime yellow. It's very prominent. We can tell, oh, okay, so this is a Xanthomonas. And not only is it white in color, you can see it actually can break down the white stuff in the background. You get those little transparent halos around the bacteria, and that tells us, okay, this one is a common bacterial blight bacterium. And then the third one is creamy white as the halo blight, the Pseudomonas one. But it has little halos. It can break down the white. And that tells us, so this is a Pseudomonas syringae. We also bacteria have little barcodes, like the Fusaria. So we also do that molecularly and then put everything in an Excel sheet and keep that to share with the breeders. And also for the bacteria, we also need to know how aggressive are the bacteria that we have currently in the North Dakota and Minnesotan fields. We do different assays. So sometimes we infiltrate the leaves just to see what the bacterium does. And you can see in our case here, this is a Pseudomonas syringae, and it kills parts of the leaf. That tells me that it's very aggressive. But because in nature, not everything is actually infiltrated, it goes by wounding, it goes by water splash. There's a lot of things by, when you have hail, you get bacteria get into the plant and start establishing themselves. And this is another way how we test how aggressive is a bacterium. And we do that by spray inoculation. So basically we just take a sprayer, like what you use to water air plants, and spray the bacterium on the leaves. And you can see here a control. So when we spray just water, there's nothing going on the leaf. Looks healthy, looks very green, very strong. But then different bacteria here give different phenotype or different markings or symptoms in the leaves. So this, for example, is the Pseudomonas syringae, and you can see that we start seeing the little necrotic spots here, and they develop over time. So we start at day one, and every day we go there and we look how much worse does it get until we can score in the end. And It gives us a little bit more of a natural idea of how the bacteria how aggressive they can be in more natural settings. Here's another Pseudomonas. They tend to be prominent here. And hold on, I have a Xanthomonas. No, I don't. You can see that also not all of them are equally aggressive, right? So we have differences. This one is, I would say, medium. This one is a little bit high, it's more prominent. And then this one is hardly pathogenic at all. And yeah, it is important that we do those assays because that is how breeders can screen their varieties. All the fungi and all the bacteria that we sample during the bean survey is actually something we keep, we take notes, and we pass on or make available to the NDSU breeding programs because, well, they're the people who actually give you the new varieties, right? And this is pretty much a wrap up for me. I would like to no, hold on. So first of all, there's something I think I need to I would like to end with, and that is I've been very honest with you, you know. In this presentation, I said a lot of times, this is a new physarum, we don't know, and that is true, we do not know. But this is really not a reason to worry. The reason that we don't know is because technology gets better and better, and we're just starting to find out things. So obviously, when you start things up, you don't know exactly at that moment, but you will find out later. And it's actually good that with improving technology, we get a better look. So actually, we're able to tell you a little bit more in detail what you have in the fields. Okay, but all of this is no reason to worry at all. I would like to also highlight that obviously I'm speaking here to you today, and I'm very grateful for that. But I'm not the only person at NDSU who does driving work, and I have amazing colleagues. For one, Dr. Markel, who's going to speak, going to be the next talk. He will tell you a lot about what he's been doing. Then Dr. Upinder Gill, he is the specialist on rest. He has beautiful data and has been working with Dr. Markel. Then we have Dr. Guping Yan, who does soybean cyst nematodes work with Dr. Markel. And then last but not least, the dry bean breeding program, Dr. Hanosono, who is my collaborator, and I'm very, very grateful that I get to work with him. And to wrap things up, I would like to thank everyone for letting us go onto their fields and sample. So in summer, when you see a couple of people in green and yellow, you know, running through the field sampling, that is us. Please drop by, say hello. We love to chat and love to talk with everyone who's interested. We are very grateful for funding from the North Harvest Bean Association. And yeah, if you have questions, please let me know.